Multi-Drug Resistance Association with Cancer Chemotherapy. He is a member of Research Society of Alcoholism, American Association of College of Pharmaceutical Science, and New York Academy of Sciences. He has honored Faculty Research Award in 2015 by TCOP. He has attended various national as well as international seminars as a speaker. I welcome you, sir, to share your experience on ABC transporters mediated multi-drug resistance associated with cancer chemotherapeutics. Over to you, sir, please.
Thank you, Dr. Rahman, and I would like to thank the organizing committee for giving me the platform to speak today about my research, and I would also like to thank Dr. Daya for introducing me to APP. So, let me. Okay, so if you can see, this is where I am. Currently, I'm working at Fullerton, and my university is in Fullerton. It's about 30 miles south of LA. And my talk is going to be on the role of ABC transporters in the multi-drug resistance that is associated with cancer chemotherapeutics. All right. So basically, what happens according to the published data, right? There are about 14.1 million new cancer cases every year, out of which 7.6 million cancer, and cancer is a disease that may actually claim about 7.6 million deaths. For all these patients, chemotherapy is the treatment of choice, and about out of the patients who get cancer every year, about 12, give or take, about 12 million new patients, they develop something called as multi-drug resistance. Now, multi-drug resistance is a phenomenon that is associated with the resistance to the structurally and functionally unrelated anti-cancer drugs. Okay, and the development of this MDR actually, it causes or it leads to the failure of chemotherapy in about 90% of patients with metastatic cancer. So brief overview of how MDR happens, right? MDR could happen because of many reasons, because of evasion of drug-induced apoptosis, activation of DNA repair mechanisms, or it may also happen because of decreased drug uptake or increased drug efflux. So in this project, what we had done is we focused on the MDR associated with increased drug efflux. Okay, and the major culprits for this type of multi-drug resistance are nothing but ABC transporters. So what are ABC transporters? These are ATP binding cassette transporters. It is a very large family, consists of about 7 subfamilies, and which contains about 48 functional members. There is one non-functional member, and these range from ABCA through ABCG. So now for ABC transporter, the members, these proteins are actually, it's a group of proteins that are either half transporters or full transporters. Now, what does that mean? What does that mean by half transporter or full transporter, right? So typically ABC transporters, they consist of 2 nucleotide binding domains and 12 transmembrane domains. Okay, now these 12 transmembrane domains and 2 nucleotide binding domains, they are divided into 2 transmembrane domains. And So, and now these nucleotide binding domain, based on the sequence similarity of amino acids, this is the place where ATP comes and binds. And ATP has a very major role, binding of ATP has a very major role in the function of these transporters. And we will discuss, about the role of ATP when we discuss the cycle of the transporter. Okay. uh, so in other words, in nutshell, what we can say that these a functional ABC transporter, it can either be a complete transporter with two nucleotide binding domains or two transmembrane domains. I'm sorry, two nucleotide binding domains and two transmembrane domains, or a combination or a dimer of two half transporters. Okay, so for example, here ABCB1, it is, yeah, and ABCC1, these are full transporters. However, ABCG2 is a half transporter. Now, what happens with ABCG2? It has to dimerize with another half transporter, right? So what happens? It dimerizes, and because when it dimerizes, it becomes fully functional, and it expels the drugs out of the system or out of the cell. Okay, some of the substrates of this transporter are topotecan, mitoxantrone, SN38, and doxorubicin. These are conventional chemotherapeutics that have been used or are being used to treat some of the types of cancers. Okay. So let's look at the proposed mechanism. Now, the reason why I'm telling you why I'm saying it proposed, because there are different theories of how these transporters work. There is no set theory as of now. But the proposed mechanism of action of ABC transporters is what happens that a ligand comes and binds to the hydrophobic pocket of the transmembrane domain, and because of which what will happen that the ATPs are recruited towards the nucleotide binding domain. The two ATP molecules, when they bind, what they will lead a conformational change in the transmembrane domain, because and what will happen that ATP will get hydrolyzed by the enzymes called as ATPases, which will actually open these transmembrane domains. Now, as the transmembrane domain is opened, the drug will be effluxed out of the cell, leading to subtherapeutic doses of the drug within the cell, because of which the drug will have no effect on the cell. So in normal physiology, these ATP binding cassette transporters, they have a protective role, and they expel the xenobiotics out of the cell, and they protect us from various kind of infections. However, in cancer cells, what happens? These cancer cells, what do? They're very smart. They actually overexpress, they exploit this mechanism of ABC transporters, and they overexpress ABC transporters on them. Now, because of which what will happen, these ABC transporters will expel or will efflux the drug out of the cell. Okay, and that will cause multi-drug resistance. Um, Now, what we did in this project, because considering that every time to come up with a new cancer chemotherapeutic that may not be a substrate of these ABC transporters, is a very time consuming and a very expensive affair. So we wanted to somehow potentiate the effect of existing chemotherapeutics uh, by actually introducing a reversal agent, by introducing an agent that may reverse this function of the efflux transporters, that may somehow block the efflux transporter, so that when the drugs are co-administered with this reversal agent, what will happen, the reversal agent will reverse the function of the transporter, and the transporter will not be able to efflux the drug out of the cell, thereby achieving therapeutic concentrations of the drug within the cell, and the drug will do its job. Now, these are some of the examples of the modulators of ABCG2. Uh, uh, you know, Fumetry Morgan C is the one that we used here because what Fumetry Morgan C does, excuse me, it is an exogenous modulator, and it blocks the function of the pump. Okay, the another one is novobiasin that we use in the study, and I'll tell you why we use novobiasin. So the objective of the project was to screen for agents that reverse the ABC transporter mediated multi-drug resistance. We went through the list of screening agents, and we found out that out of these agents, there is one drug that is AC220 at 3 micromolar. It showed promising effects, reversal effects on ABCG2. Cell lines that cancer cell lines that were overexpressing ABCG2. Okay, so a little bit about AC220. AC220 is also marketed as quizartinib. It is a potent and selective inhibitor of FLT3, which is a validated target in the treatment of AML. It has already passed phase 2 clinical trials for AML, and it is very highly effective as a single agent, can be combined with chemotherapy without additional toxicity. So, so we hypothesized that AC220 would reverse the ABCG2 mediated multi-drug resistance. Okay, we to prove our hypothesis, we had a few specific aims. We wanted to determine whether AC220 reverses ABCG2 MDR, and we also wanted to determine the mode of interaction of AC220 with the ABC transporters, and we wanted to see how does AC220 impact the growth of tumor in the athymic nude mice. Okay, so before we started the study, we wanted to check the expression of ABCG2 in all the cell lines that we were using. So we confirmed the expression of the protein with the help of Western blot, and as you can see that the protein is present. ABCG2 is present in H. So we took the cell lines that we took were H460MX20. This is the drug selected resistant cell line that is made from H460 cells. So H460 are the parent cells, and H460MX20 is the drug selected cell line. Then we also took HEC293 cells that were transfected with empty vector, and HEC293 cells that were transfected with variants of ABCG2. So ABCG2 482R is the wild type ABCG2. Uh, However, ABCG2 482G and ABCG2 482T, these are the mutant ABCG2s. Okay, and then we also, excuse me, we also worked on one specific drug selected cell line, S1M180, that overexpresses the ABCG2 mutant, G mutant actually. Okay, so then we went ahead and did the cytotoxicity studies, and in the cytotoxicity studies, we saw that quizartinib alone, had no minimal or none to minimal toxicity to all the cell lines that it was tested on, and with the IC50 values of more than 10 micromolars. Based on these studies, actually we selected two doses to work with. One was 0.75 micromolars, and the other one was 3 micromolar. So now we wanted to find out whether ABCG. We wanted to do a preliminary study to see whether ABCG2 is the effect of ABCG2 is being reversed by combination, by the treatment of quizartinib. So we see that here in this panel, we see that HEC293, we have taken HEC293 cells transfected with null vector, HEC293 cells transfected with uh, uh, ABCG2 wild type protein, uh, and ABCG2 G mutant and ABCG2 T mutant protein. Okay, so we can see that here quizartinib significantly increased the cytotoxicity of mitoxantron, SN38, and topotecan in wild type and T mutant ABCG2 transfected cells in a concentrated dependent manner. However, quizartinib showed only a very moderate reversal effect in 482 mutant, 482G mutant. Uh, Now we can also see that here you have used novobiacin. Novobiacin is actually a known inhibitor of G mutant and T mutant ABCG2. But And we can see that the reversal effect of quizartinib on T mutant is better than the novo effect of novobiacin. Okay, uh, moving on to the drug selected cell lines, if we can see that we saw the similar effect on drug selected cell line. Now this is H460MX20 that overexpresses wild type ABCG2 protein, and we can see that then we also wanted to check the effect of quizartinib on the drug selected cell line that overexpresses G mutant. And if you see here, it corroborates with the results that we got from the HEC293 study. So here also, quizartinib very moderately sensitized the G mutant cells. Uh, 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 and in all the studies, needless to say, quizartinib did not sensitize the parenteral cell lines. Okay, so so now we wanted to understand the mechanism of reversal. What is the mechanism? How is it happening, right? So we examined the effect of quizartinib on the intracellular accumulation of ABCG2 substrate anti-cancer drug, that is tritiated mitoxantron, in ABCG2 overexpressing cells. The intracellular concentrations of tritiated mitoxantron, it was measured in cells with or without quizartinib. Quizartinib at 3 micromolar significantly increased the intracellular accumulation of tritiated mitoxantron in both wild type ABCG2 as well as T mutant ABCG2 transfected cells. But it moderately increased the intracellular concentration of mitoxantron in the cells that overexpressed 482 G mutant AIA. Okay, so now this increased intracellular accumulation of mitoxantron by quizartinib, it may be happening because of two mechanisms. One is that probably quizartinib may increase the mitoxantron uptake, or another possibility is that quizartinib may inhibit the efflux of mitoxantron. Okay, so within the realms of our lab, what we did, we performed an efflux assay and found that the extrusion rate of treated mitoxantron uh, was significantly inhibited by the combination of quizart in the combination group of quizartinib and I'm sorry, was significantly inhibited by quizartinib. And this was again consistent with the data that we have been getting, that we could see the effect on the wild type ABCG2 as well as T mutant, but we did not. We saw a very moderate effect on the G mutant ABCG2. So now the reversal of ABCG2 mediated MDR by quizartinib, it could occur because of uh, two mechanisms, either by inhibiting the function of the transporter or by down regulating the expression of ABCG2. So to analyze the effect of quizartinib on ABCG2 cells, what we do, what we did, we incubated the cells with the drug, the cells that were overexpressing ABCG2 with quizartinib at 3 micromolars for about 24, 48, and 72 hours. And here we see no change in the expression of the protein. So that means that the quizartinib does not downregulate the ABCG2 in cells, but it rather inhibits the function of the efflux function of the transporter. So then we wanted to see the effect of quizartinib uh, on the rate of growth of tumor in athymic nude mice, and the weight of tumor actually. So what we did is we in this slide I'm showing you the effect of AC220 on MX20 xenografts. So what we did, we injected the mice with H460 cells, and in one arm and the other arm, we injected the mice with AC H460 MX20 cells. Okay, and then we actually treated the mice with either saline or AC220 alone, because we wanted to see the effect of AC220, whether it is having any effect on the tumor or not. And we also treated we had a group that was treated with topotecan alone, or and also we had a group that was treated with the combination of topotecan, that is an anti-cancer drug, and AC220. AC2 the dose of AC220 that we took was 30 milligrams per kilogram. So if you see over here that the tumor volume actually significantly reduced in the combination group. Not only tumor volume, but the weight of tumor was also reduced in the combination group. So now here looking, this slide talks about the effect of AC220 on H460 xenografts. And here, if you can see that the combination group was not different from the topotecan alone group. So thereby saying that because this H460 xenograft had no ABCG2 expression, so that goes well with our with the uh, with our study. Now to see whether AC220 had any, or the combination of AC220 and topotecan had any effect on the tumor weight. Here, if you can see that the combination had it was not significantly different from the topotecan alone group. So and that is what we expected. Now, we also wanted to see whether the combination had any toxicities on mice. So here we can see that, and toxicity we measure by the help of body weight. So we can see that the combination treatment had no weight loss. So that is an indicative that the mice didn't, I mean, the combination had no toxicities. Okay. So here with this study, we proved that AC220, it reverses the MDR associated with ABCG2 wild type and T mutant. Um, and to draw the conclusion from this study, Actually, these are the conclusions that AC220 reverses the ABCG2 mediated MDR in vitro as well as in vivo, and with associated with wild type ABCG2 as well as T mutant ABCG2. AC220 significantly increased the intracellular concentration of mitoxantrone by decreasing the efflux, and it may be combined with other conventional anti-cancer drugs for improving the chemotherapy, and it may improve the chemotherapy in patients that overexpress the wild type and mutant ABCG2. With that, I would like to acknowledge my collaborators, Lou and Jason Chen, and the students who helped me with the project, Nagaraju, Chris, and the two amazing high school students who worked with me on this project, Kanav and Lulu, uh, for making this project possible. And currently I'm working here at Marshall B. Ketchum University. This is the amphitheater of our school. With that, I would like to say thank you.
Thank you, Dr. Priyank Kumar. Thanks for your wonderful lecture. And I'd like to request you to please. Yeah. Now, moving upon the next speaker, I am having in the panel that Dr. Rakesh Kumar Tiwari from Chapman University, California, USA, who is going to talk on the development of amphipathic cyclic peptides as antimicrobial agents against resistant pathogenic bacteria. So I request one of my organizing committee colleague to recite the profile of Dr. Rakesh Kumar Tiwari and request him to start his presentation.
Thank you very much, sir. Dr. Rakesh Kumar Tiwari is currently working as Assistant Professor of Biopharmaceutical and Biomedical Sciences, Chapman University School of Pharmacy, Chapman University, California, USA. He completed his education from University of Delhi. His major research interest in drug design against multi-drug resistant escape pathogens, drug delivery using peptides, drug design for neurotherapeutics, development of synthetic methodology. He was a senator in Chapman University from 2013 to 2017. Presently, is a member in the Science Safety Committee of Chapman University. He graduated from these students. He has in 2021 he has been awarded for the mentorship of undergraduate research and creativity activity by Chapman University, undergraduate research fellowship, postdoctoral fellow research excellence award, and many awards have been awarded by various societies and various associations. He is the member of American Chemical Society, USA, member of College of Pharmacy Professional Research, member of American Association of Colleges of Pharmacy, American Association of Pharmaceutical Scientists, member of American Association for Advancement of Science, American Peptide Society, Asian Council of Science Editors. He is also a reviewer of various grants and scientific journals. Presently, he is a chief editor of International Journal of Medical and Pharmaceutical Case Reports, associate editor in Pharmacologia, guest editor in Special Use of Molecules, guest editor in Special Issues of Pharmaceutics, editorial board member for Central Nervous System Agents in Medicinal Chemistry. More than 100 articles have been published in reputed national and international journals with the H index of 32 and edited 2 book chapters and various publications and conference attended and poster presented. Now I would like to welcome Dr. Professor R.K. Tiwari sir for delivering the 4th lecture on the topic development of amphipathic cyclic peptides as antimicrobial agents against resistant pathogenic bacteria. Over to you, sir. Please unmute yourself.
Thank you
Thank
so
you.
much, Doctor. Thank you so much, Dr. Singh. Can you hear me?
Yes, sir. Yeah. You are
Thank you
on.
so much, Dr. Singh, and thank you so much to organizing committee to give me this opportunity to share my research, and thank you to Dr. Dhaya to connect me to APP. I'm so excited and very humbled to be joining this group of eminent scientists around the world, and I'm so happy to share my research. So let me share my slides, and then
Yes,
is
sir.
my
Set
slides are
now.
visible?
Yes, sir.
Set now.
Yeah, okay.
All set.
Thank you so much. Sure. So this is the place, Chapman University School of Pharmacy. It is in South California. It's about 40 miles away from Los Angeles. And you might all hear about the pandemic right now in LA is very much, but we are very far away from there. And this is a new campus. It's built in 2013 when I started my career at Chapman University. And before that, I started in a Stu Nather University in Greater Noida for a short time. And then I moved to the this new pharmacy school. So this is a overarching area of my research. So basically my research is an area of medicinal chemistry, and I love to use peptides for any purpose, any function, including drug delivery. For example, using cell-penetrating peptides and using peptide for purpose of neurotherapeutics, using protein using protein-protein interaction as a drug targets, and also to deliver some chemotherapeutic agents using biomarker expressed in cancer. So that area is very much in demand, tumor targeted drug delivery. And the another area where I'm very excited to follow the natural, natural instinct, for example, following the nature, how the plants, animals, they have a protective role to protect themselves from the bugs and other kinds of pathogens. So they all excrete a secondary metabolites or compounds named as antimicrobial compounds. So those are all from the natural resources. And here in my lab, I am pretty much trained as a chemist, like synthetic chemist, medicinal chemist. So I follow the nature's nature's examples to develop and optimize some compounds in my lab to use them for various applications. So today I'm going to talk about 3 or 4 different projects based on the time, and hopefully I may able to share you some of the data I generated at Chapman University when I started my independent career. And this is related to this antimicrobial agents. So everyone knows about, the problem of antimicrobial resistance, antimicrobial drug resistance. So as you see in this cartoon, you may see that the antibiotics are no more working because bacterial system is developing resistance. And this is a huge problem coming up. And there are also these bugs, they are especially found in the hospital settings, and they are known as superbugs. And this is a series of 6 bacteria named as escape pathogens, and they are very prominent in the nature. And they are found in the hospital settings. Any type of antibiotics you use it, they are not effective. So here we are going to show you the data generated in the lab about optimization of a lead compound. We try to optimize in the lab and test it in the test it against these resistance pathogens to develop a new generation of antibiotic. So this is the outline. There are like three projects I want to share, if time permit. And I am going to start with the introduction and then hypothesis, then methodology, results, conclusions, and followed by the acknowledgement. So as I explained, the area of infectious disease is developing resistance to the known antibiotics. The reason is that people are using, excessive amount of antibiotics. Either they need it or not, or they know what kind of antibiotics need to use. So this overuse of the antibiotics, created the problem of antimicrobial resistance. And there is a good example I want to share all of you, is that there was a global study in United Kingdom government, sponsored that with the Wellcome Trust to try to find out about this problem of antimicrobial resistance infections around the globe. And this agency found out that by 2050, if we don't cure this problem of antimicrobial drug resistance, the annual mortality would be 10 million people every year. So this is a huge problem. And some more data I can share you from CDC, the Center for Disease Control and Prevention at United States. According to their report, more than 23,000 people die annually from antibiotic resistance bacteria infections. And more than 2 million people get infection because of these specific bacteria, which are not responding to the regular or classical antibiotics. And this whole problems contribute about $55 to $70 billion of economic impact. And because of all these issues, in United States at White House, they developed a program known as a combating antibiotic resistance bacteria to develop some newer generation of antibiotics or to overcome this problem. So there are series of the reports suggested that the escape pathogens, and escape is the name. It is for E is for Enterococcus faecium, S is for Staphylococcus aureus, K is for Klebsiella pneumonia, A is for Acetobacter pneumoniae, and P is for Pseudomonas aeruginosa, and E is for Enterococcus bacteria. So these 6 bacteria are categorized in a class of escape pathogen because they escape from the antibiotics, and all kinds of antibiotics you use it, these bacteria are showing resistance against that. So there are a lot of efforts going on to develop newer generation of antibiotics to deal with these escape pathogens. So one of the class of the compound is reported from the nature, and this is an antimicrobial peptide, and they are produced from the eukaryotic organism as a part of their innate immune response to bacteria or fungal pathogens. And they have unique mechanism of action. So they are considered as a unique class of antibiotics, and they can be used and optimized to develop a new class of potent antibiotics, which can be effective against these escape pathogens. So in brief, this class of peptide, antimicrobial peptide, acronym is AMPs. They are representative peptide. They consist of about 5 to 50 amino acids. And their target is basically bacterial membrane. And they try to rupture the bacterial membrane. And because of that, bacteria will die. And these class of peptides are because they are working on the bacteria, right? A bacterial membrane. So because they disrupt these lipid bilayer structure, and because of that, this class of compounds can be working on a broad spectrum of bacteria. And another very important property for this class of compound is that these antimicrobial peptides has a less chance of developing resistance because bacteria has to do several rounds of mutations to develop new type of, phospholipid bilayers. So the chances of resistance is very low. And there are several, compounds or, peptides from this class, such as a human cathelicidine peptide, LL37, LL16, bricolacidine, pexigenin, dalmatidine, navaxidine, OP145, omeganin, melatonin, meganine, so on. There are several of these compounds, these peptides from this class of antimicrobial peptides are in the clinical trial. But all of you will be surprised that there is only one drug named daptomycin that is got approved from the FDA, but there are so many of them, they got either some kind of issues related to the toxicity, and they did not, successful for, clinical trial. So as I mentioned, the advantage of this class of peptide is that they are broad spectrum antibiotics. They can kill the bacteria because they are bactericidal in activity. They have low chance of developing resistance, and they also have some other activities such as anti-inflammatory activities. However, they have a disadvantage, such as a cost of discovery and screening is high. They are cytotoxic because sometimes these peptides or these compounds cannot differentiate between mammalian cell versus bacterial cells. And a lot of time these compounds have a lot of cationic amino acid, like for example lysine, arginine, and these cationic amino acids are sometimes these compounds are not stable on salt, serum, or pH. And also because these are peptide, so peptides has a lot of issues with pharmacokinetics and pharmacodynamic issues. So these are the disadvantages. But overall, this is a unique class of compound. If optimized, very purposefully, they can be a good antibiotics. So there are some examples from the literature. You may see that daptomycin is here. They are cyclic peptide, and you can see they have a fatty acid chain. So they are also having some hydrophobic amino acid, hydrophilic amino acid, or cationic amino acids. So they are amphipathic in nature. Here is a polymyxin B or cholecystine, right? So they are also, you may see they also have a hydrophobic chain, right? And, some cationic charges. Same here, gramicidine, gramicidine S. So they have, the cyclic structure, and here is a vancomycin. So all these shows a good example of, good examples that this class of, compounds can be a good antibiotics. So now coming to the lead compound. So this is a cyclic peptide containing 8 amino acid. Here is a tryptophan. You can see 1, 2, 3, 4 tryptophan. Here is a 4 arginine. And this is a cyclic peptide. And this peptide was found early 2014 that this peptide is active against MRSA, methicillin resistant Streptococcus aureus, with MIC is 2.97 microgram per ml. And this peptide also shows synergistic activity with the tetracycline. So based on this activity, there was some more SAR, structure activity relationship was performed. And you may see that this is 4 arginine, R is for arginine, W is for tryptophan. And you can see this shows a good activity against gram positive, but from gram negative, it doesn't have much activity. However, the linear part, like this is a linear peptide, you can see that linear peptide is more better for gram negative. So the effort was made to increase these number of amino acid like arginine and tryptophan, and we try to do some structure activity relationship. We try to change some other amino acid like glutamic acid, like positive charge, negative charge, and then added fatty acid chain, small chain, bigger chain, or added some tryptophan, some hydrophobic residue, try to improve the activity. But during this time, we had a very short, optimization in the hand, and we did not find that the activity improved. So based on this understanding that this compound has a good, MIC value for gram-positive bacteria. So when I started my career at Chapman University, I said, okay, let's, optimize this cyclic peptide and try to see if we can get a better activity and more better compound. So we hypothesized that if a peptide has the appropriate hydrophobic and positive charge residue, they have potential, antibacterial and synergistic activity with other antibiotics. To take that mind, we wrote some objective again. As we work with the students, we have to write down all the plans. So we write down the objective that we'll make and synthesize these peptides and then culture the bacteria, these resistance bacteria in the lab, and then try to find out the MIC value, try to establish the structure activity relationship. So here is the design part of the library. So we want to change this X. This X was earlier tryptophan. Now in place of tryptophan, we try to use leucine, isoleucine, alanine, phenylalanine, and tyrosine. And then in place of this, arginine, we try to use lysine and glutamic acid. And we also increase more tryptophan, more arginine, and we also try to change, different stereochemistry. For example, D version of amino acid, methylated tryptophan. We try to see if we do these modification, if we have a better activity or not. So this is again, a scheme to how to synthesize these compounds. So pretty much, we use solid phase ACMOCT, we use solid phase chemistry. These amino acids are assembled. And again, this is a very straightforward, synthetic routes. And once we assemble all the amino acid, then we cleave the resin selectively and then do a cyclization, remove all protecting group, we get this cyclic peptide. So we synthesize all this series of peptides, and you will be surprised to see that this is our original peptide. And these are the modification from, tryptophan to tyrosine, phenylalanine, leucine, isoleucine, alanine, glycine. None of them work. It was very surprised. None of them shows any good MIC, or even not similar like as the parent one. And these are the linear ones. And here is the other series where we change, arginine, right? So we change arginine to glutamic acid and lysine. Here we used methylated tryptophans. MIC became twofold, right? So it is from 4 to 8 now. Here is the D-arginine, again, not much change. It's a loose potency basically. And when we change the more arginine or more tryptophan, again, we did not see any better results. So overall, again, after that, we try to see if we have, cytotoxic compound or better compound. So here we have, these are 2 cancer cells, and here is the normal cells. And we found that, they are pretty okay, not much toxic, up to 10 micromolar. You can see it up to 10 micromolar, they are okay. So they are the 3 different cell line. Up to 10 micromolar is fine, but at 50 micromolar, they was toxic. So from this first SAR design, we conclude that this series of peptide, already, like for example, R4W4 is still a optimized peptide. There is a requirement of tryptophan, cyclic part is important, stereochemistry is important. We need L configuration of the amino acid. So based on that, we find it out that, this parent compound is optimized peptide R4W4. And then we design another project where I want to, conjugate another antibiotic, for example, levofloxacin, with this cyclic peptides and try to see if we get a better antibiotic, properties or synergistic activities. So here we try to design a project, and we take another antibiotic. This is a Q-levofloxacin. It doesn't have this piperazine motif. This is a part of pharmacophore. Without this ring, levofloxacin has no activity. This is the compound without this piperazine ring. So we take these 2 compounds and try to conjugate with our cyclic peptide. And all of you know levofloxacin is very important antibiotic. It works against both gram-positive and gram-negative bacteria. And that's the reason I thought, okay, I will synthesize a conjugate and which will have a better activity. So this is a chemistry, right? The synthetic scheme, we attached this levofloxacin on the solid phase, then did the cyclization. This was pretty easy because if you go back, you can see this has this carboxylic acid group. And because of this carboxylic acid group, it's easy to attach it on the solid phase peptide chemistry. And then we cleave it, and cyclize, cleave it, and, deprotected. We got these two conjugate. And this is the structure. You can see here is a levofloxacin part, and here's the Q levofloxacin part. So these two conjugates was synthesized. And very interesting is the data. You can see these are the control antibiotics. This is a Q levofloxacin. Without piperazine motif, it doesn't have the activity. And this is the cyclic peptide again against MRSA4, and here is a KPC is a 16. And this is the conjugate one with levofloxacin, and this is a linear conjugate. This is a cyclic peptide conjugate. You can see from 4, we come to 32. Very surprised. We lost the activity basically. And here from 16 to 128. And this is again, a linear one is more bad. And this is the physical mixture. When you mix these, antibiotic and cyclic peptide is 1:1. So what we saw that, we saw that this is, the physical mixture is better than the conjugate. And this was kind of disappointing results. And what we thought that it might be because this conjugate has to, hydrolyze. The conjugate has to hydrolyze and release those antibiotics and cyclic peptide to get the activity. Maybe this conjugate is not hydrolyzing. That might be the reason. So this was the conclusion from this second project. And in the third project, I got a master's student, and he was very excited. He said, no, I want to work on this project. I said, okay, look, we can optimize this peptide by doing some fatty acylation. So we thought, okay, when we do a fatty acid chain, it may enhance more interaction with the membrane, and this peptide will be more better agent. So this is our cyclic peptide. We try to add this lysine, and from this lysine, we can do different fatty acylation on the lysine amino group, the side chain amino group. And we can go with the, different series of R group. So on that basis, we designed the compounds, we designed the scheme, right? We synthesize again on the solid phase. The chemistry part is not a problem. And we use different fatty acid anhydrides. They are commercially available. We just did a reaction, and we can conjugate different types of fatty acid chain in this series. So these are the one, and from C2, C4, C6, C8, 10, 12, and 14. So, and we went up to C20, but the solubility was a problem and purification was a little challenge. So we only tested up to C14, C12. So this is the MIC determination. Again, here is a MRSA, right? Here MRSA is 4. This is our parent compound. And you will be surprised to see that once we added a lysine, the activity decreased by 4-fold. And then we start doing acylation, C2, C4, C8. Then again, activity was bad. But when we came up to C10, the activity came back to the parent compound. And again, at C12, the activity again dropped by 4-fold. So this was very interesting observation that after doing the fatty acylation, these peptides lose the activity. And this might be related to the structure, this fatty acid chain, which might be making this, this peptide interaction with the membrane difficult, or maybe this fatty acid chain is trying to protect this, these tryptophan residues for interaction with membrane. So that might be the reason. Anyhow, then we try to do some cytotoxicity studies. And here you can see this is a cell viability, this is SKV3 cells, this is a CCRF, and here is a HEK cells. And you can see up to, they are little bit, this is a normal cell, up to like 10 to 10 or 15 micromolar, they are okay. After that, they are all cytotoxic. As I mentioned that this series of peptide has interaction with the membrane, and that might be the reason they are showing cytotoxicity. So we try to do, usually hemolytic assay in the lab. We purchase the red blood cells from the vendor and try to do this simple experiment. And here is the daptomycin. This is a standard for us to follow up. Here is a polymyxin, and here is a cyclic peptide R4W4. You can see the SC50 means the 50% hemolysis of the blood cells. Looks like, it is not better, like they have some cytotoxicity. They are not better than daptomycin and polymyxin. So on that basis, we also see that fatty acylation is a little bit more better. They are, about similar like, daptomycin, or in the range of daptomycin. So on that basis, we also did some more cytotoxicity studies for these series of peptide by some other labs, so that we are very confident our results are reproducible. So you can see that they are very safe on other cell lines. This is a liver cells. So this was a little bit positive, and this was a little bit positive. And here is another study we tried to do in vivo toxicity to make sure these these peptides are safe for the animals. So we used CD1 mice, and we gave them a different doses, like 5 milligram, 10 milligram, 25 and 50 milligram per kg. And you'll be very happy to know that most of these animals was healthy. They don't have any changes in the liver or spleen. This is a histopath data. So this peptide did not show toxicity. So I'm at this label from this project, and we are looking forward for a efficacy studies in the animals. So this is the conclusion part. And now, again, from this series of experiments, we conclude that our cyclic peptide has a potential to develop as a antibiotics against gram-positive bacteria. And I want to now, acknowledge my lab students. These are my students, a series of students, 2 masters, 1 PhD students, and few undergraduate. And here are the collaborator at my institute. Dr. Yamaki is a microbiologist, and Dr. Parang is my old mentor and collaborator. So we all develop all series of, SAR. And funding from this school and this AJK Biopharmaceuticals, who are trying to push these compounds for efficacy study. And most likely the plan is to go for future development of this antibiotic. Thank you so much for your attention, and thank you so much.
Thank you, sir. Thank you for your very good lecture on this topic of antimicrobial agents. So with this, we have completed all the invited lectures, and we have one more invited oration in this session to complete. And for that, I would like to invite Dr. Gavaskar Terry Ramanand Singh to please be on stage, and my colleagues to invite him for his deliberation.
Thank you, sir. I would like to recite a brief profile to Dr. Terry Gavaskar Ramanand Singh. He is presently working as a professor in the University of the West Indies. He did his PG, UG, and PhD in biochemistry from the University of West Indies. He published several articles in various reputed national and international journals. He has been awarded various grants and awards like Distinguished Medical Representative and the Medical Sciences Student Council in 2018. I welcome you to share your research experience on the sex-independent angle of type 2 diabetes. Over to you, sir.
Okay, thank you very much for that introduction. And I would like to thank the APP for having me here and inviting me to this session. And I would also like to thank the presenters before for such amazing presentation. So I would like to get into my topic for today's session, which is entitled The Sex-Independent Angle of Type 2 Diabetes. So let me share my lecture slide with everyone. So most people would know this individual. Her name is Kim Kardashian, or Kim Kardashian West, and she's a very famous reality television superstar in the television series Keeping Up with the Kardashians, and is the individual who is dressed in white. And critics and admirers of Kim Kardashian will tell persons that she's well known for a curvaceous figure. So what I'll be doing with Kim Kardashian here is actually placing her into this equation. So, and I'll get more into what these letters, etc., represent. I mean, you could already see the pi, you can see the tan inverse function. And so I'm going to place Kim Kardashian into that specific equation. And I'll show you how I'll do that later. But let's consider the literature regarding the sex-independent angle of type 2 diabetes. So when you look at the literature, you get these two main points when considering obesity and type 2 diabetes, that waist size and waist-to-hip ratio are superior to body mass index in characterizing central obesity, and that waist size and waist-to-hip ratio are more highly correlated to glycated hemoglobin and fasting blood glucose as compared to that of body mass index. And the number of articles that I sifted through were approximately 125. But I'll give you a synopsis of those articles. So let's look at in 2009, here we have a review article. 44 articles were reviewed, and it showed that elevated serum glucose was predicted by increase in waist-to-hip ratio and body mass index. And the author, Friedel, was able to conclude that a measuring tape provides the most useful and cost-effective measure for the prevention of type 2 diabetes. Not a specific drug, but just a tape measure. Now, we have Kiawa Nyam-Dodge. They reviewed 52 articles, and they found that waist circumference, which is represented by WC, and waist-to-hip ratio, which is represented by WH, are better discriminators for type 2 diabetes as compared to body mass index. And in 2015, Papetas and his colleagues, they reviewed 233 articles, and they found that central obesity, not obesity for the entire body, but that central obesity in the abdominal area is more prevalent in patients with type 2 diabetes. Now we were wondering, okay, so we're in 2015, but what about articles that are more recent? So in 2018, Skuberg and his colleagues, they published an article in BMJ Open, and they demonstrated in a cross-sectional study the superiority of waist circumference, that of waist-to-hip ratio and body mass index for type 2 diabetes discrimination. We have in 2020, published this year in April, in this article called The Pathophysiology of Type 2 Diabetes Mellitus in Non-Obese Individuals, an overview of the current understanding. Now that article tried to delve into patients with patients with type 2 diabetes who are non-obese. But again, from their studies, they saw that type 2 diabetes is much more prevalent in patients who are obese and who have central obesity. So considering all the literature and trying and seeing that there are some problems in it. So one of the three problems are this. So one, there's difficulty in exactly defining central obesity with an anthropometric parameter. Some of you may know defining it using body mass index, but there is difficulty in defining central obesity. there is no general agreement on which is the best predictive anthropometric parameter for type 2 diabetes. So from the British Journal of Anesthesia, volume 116, published in 2016, it says that there are limitations of BMI as an obesity measure for perioperative risk, and BMI is the most commonly used measurement for assessing obesity in adults, but has limitations because it does not distinguish overweight due to excess fat mass from overweight due to excess lean mass. So there is a problem with this BMI characterization. And our third problem, when you consider all the literature, is that there's no anthropometric parameter that surpasses the diagnostic capability of HbA1c for the condition of type 2 diabetes. So HbA1c, in a sense, reigns as the king of diagnosis for type 2 diabetes. So we have these questions. Can an inexpensive and better model for central obesity actually be developed? And will the output of this model be related to diabetes? Can our goal be to actually surpass the diagnostic capability of glycated hemoglobin HbA1c? So considering these research questions, my research focused on these objectives. It was one, to develop a novel sex-independent anthropometric index that's related to type 2 diabetes. The second objective is to examine the superiority or possible inferiority of this novel index as compared to other anthropometric indices when related to type 2 diabetes, and to look for possible correlations with biochemical blood parameters such as HbA1c, which is glycated hemoglobin, and FPG, fasting blood glucose. Also could be represented as FPG, fasting plasma glucose. So in this research study, an equation, a model was actually developed in order to characterize our central obesity and to see if it was related or not related to type 2. I mean, we are hoping for the best with our model. So in our equation, our model development, we applied Euclidean geometry to waist and hip measurements. And specifically, in that Euclidean geometry, it stems from a derived trigonometric relationship among waist and hip measurements. And if you recall from the equation, you saw that trigonometric relationship there where we use the tan inverse function, or tangent inverse function. And angle index is the terminology. So we use the term AI to describe our novel index. So the hypotheses were developed in this study were: one, patients with type 2 diabetes have a higher angle index as compared to the controls. The second hypothesis is AI is a superior sex-independent anthropometric index as compared to other sex-independent anthropometric indices in relation to type 2 diabetes discrimination. And AI has the highest correlation with HbA1c and fasting blood glucose as compared to other sex-independent anthropometric indices. So these are the hypotheses. Now, when you actually perform the test, it can refute these hypotheses. And you may be wondering why it is that we are trying to choose a sex-independent anthropometric index. Now, you have to remember that waist-to-hip ratio, there are different cutoff values when applying to males and females. But we want something that is similar to HbA1c. So when you're using HbA1c in diagnosing type 2 diabetes, there's only one cutoff value. Well, it depends on your region. But most regions would use that cutoff value at 6.5%. For males and females, there's no difference. But if you compare that to waist-to-hip ratio, there are going to be different cutoff values for ill health, whether it relates to type 2 diabetes or some form of obesity when considering men and women. But we want something that is sex independent, does not depend on the sex. So our inclusion criteria for this research study were patients with type 2 diabetes, it's an obvious, controls without type 2 diabetes, and patients over the age of 18 years. We, of course, included alcoholics, persons having any form of cancer, those with liver disease, thyroid or parathyroid hormones, patients who were pregnant, and patients under the age of 18, as well as smokers, as they could interfere with the results and our analysis. Now, for the controls, we strictly restricted persons who had an HbA1c value of equal or greater than 6.5%, and we ensured that our controls, they did not exceed a fasting blood glucose or fasting plasma glucose of above 120 milligrams per 20 milligrams per deciliter. We obtained ethical approval from the University of the West Indies, the North Central Regional Health Authority of Trinidad and Tobago, as well as the Southwest Regional Health Authority. Now, the sampling that was done is called convenience sampling. So those who are attending the clinics, we would give them information about the study being conducted, and they decided whether or not they would want to participate in the study. The sample size when we started was 121, and then the subjects had to fast before blood draw. So this is initially when we did the study, but we actually there were certain entities and organizations that doubted these results, so we had to make the size larger and repeat the study in order to, to develop a convincing argument. But if you use the sample size of n is equal to 121, our total sample size, you have a very high power. The power of the study was 95%. So using n is equal to 121 is very good for this particular study. We obtained, of course, the patients, their height, their mass. We measure the patient's waist at 2 locations and the hip size at 1 location. So when I say we measured the waist at 2 locations, so the enhanced protocols, which is the National Health and Nutrition Examination Survey, they have a different protocol as to where to measure the waist as compared to the World Health Organization protocol. So we measured the waist at 2 regions. So one was at the umbilicus, and one was just above the umbilicus, following the World Health Organization protocol. So those were 2 sites in which we actually measured the waist. And I have a diagram to actually give you a visual representation of where we measured those waist size. But we only measured the hip circumference at one region. The hip circumference was measured as the largest circumference below the umbilicus, passing around the gluteus maximus, but not extending beyond the gluteal region. These blood samples that we collected were centrifuged at 2000 G, and we stored it at those samples, the serum samples, plasma samples, were stored at minus 70 degrees Celsius before we analyzed it. So so in our research methods, the lipid profile was examined, fasting blood glucose, our glycated hemoglobin, we determined, all using a dry chemistry analyzer. And in this project, we also had a separate project looking at the vitamin D levels, the C-reactive protein, and we also did DNA extraction, because we collected all these samples. So instead of repeating the study over and over, we already had blood samples. So this, the overarching study was looking at type 2 diabetes patients, comparing it to controls and looking at this angle and X, but then there were certain sub-projects that came out of that. And since we already had the patient's blood, we obtained ethical approval for these other projects, looking at the vitamin D levels and C-reactive protein, and also examining the DNA. Of course, those results are not reported here, as we are just looking at this novel index I've called angle index. So let's look at the parameters considered in the study. So we looked at fasting blood glucose, as I mentioned before, HbA1c, triglycerides, cholesterol, high density lipoprotein, low density lipoprotein, and very low density lipoprotein. That will make up our lipid profile. We got information on patients' age, mass, height, blood pressure, their minimal waist circumference, the umbilical waist circumference. So the minimal waist circumference, remember that follows the World Health Organization protocol, is very similar to that. And then we have the umbilical waist circumference, so where you would put the measure the measuring tape around the patient's umbilicus. So normally the minimal waist circumference is going to be higher in the person's body region as compared to the umbilical waist circumference. And of course, we measure the hip circumference, and we determine waist to hip ratio, minimal waist to hip ratio, the body mass index, conicity index, abdominal index, and finally here the angle index. And all these indices are ways scientists try to use to really understand or to get a mathematical representation or an index of what central obesity actually is. These results were analyzed using these statistical packages. We use G-Power, Minitab 17, as well as IBM SPS version 22. So what is the geometrical interpretation of this, this novel angle index? So you can see it here. So you can just picture your umbilicus. So if you just picture your umbilicus in your mind, so each circumference is flattened out into a horizontal line, which is actually parallel to the other lines. So the this dimension here, the DMU, can be visualized as the perpendicular distance from the transverse section of the minimal waist region, so minimal waist region, right, to that of the umbilical waist region. And then DUH, right, can be visualized as the perpendicular distance from the transverse section of the umbilical waist region to the transverse section of the actual hip region. So you simply look at these measurements are very simple to take in a sense. So you look at the umbilical waist, right? So where your umbilicus is, you get that measurement, that circumference, and then above that you're going to have you'll have this sink or this natural dip that we actually have as humans, and we call that our minimal waist. We also measure the hip circumference, which is below the umbilical waist. And so you can understand what each of these letters mean. So UC is going to be our umbilical waist circumference, HC is going to be our hip circumference, and WMC is our minimal waist circumference. And so we just plug these values in here and we are able to get our angle index. So we have pi, and then we use a tan inverse function when we do our divisions within here. Now, there was one modification that we made here. We did not measure DMU and DUH at the initial stage of our studies. What we did, in future studies, we want to actually measure that. What we did was we assigned DMU and DUH as a value of 20 centimeters. And this AI value is in radians. And of course, you can just convert it to degrees. So 1 radian is equal to 57.3 degrees. So some persons who don't like to work with radians, that is a way you can represent an angle. You can use degrees. So in this interpretation here, we have our minimal waist here as 104.1 centimeters, our umbilical waist here as 96.8 centimeters, and our hip circumference here as 114.5 centimeters. So when you put these values together, what angle do you get? So you plug that in the formula. Of course, your DMU and DUH is going to be 20, so 20 by 2 is 40. And the answer that you get is going to be 145.7 degrees. So let's put now Kim Kardashian into the equation. So which of these models do you think is Kim Kardashian? Is it your top left, your top right? Is it the one at the bottom, or the bottom right? Bottom left or bottom right? So if you guessed the top right, that would actually be Kim Kardashian West. So here we have our angle index, and we compared between that of males and females, and we realize that is actually the same. But if we look at that with the controls, the controls and type 2 diabetes, we see that patients with type 2 diabetes actually have a higher angle index as compared to that of controls. And we want to know which one is actually better because we're considering the sex-independent angle. So remember, we have other parameters that are actually going to be significantly higher than patients with type 2 diabetes. But we want to know if angle index can actually compete with that. So what we would do, we'd apply a receiver operating characteristic curve. And you can't see it here if angle index is the true winner. So what I did was actually put the values here in this nice bar chart so you can actually see. And you can actually see here that angle index has the highest area under the curve. So thereby it indicates clearly that it is superior in discriminating type 2 diabetes. What about its correlation with HbA1c? Again, it has the highest correlation as compared to all these other indices. Let's look at fasting blood glucose again. Again, it has the highest correlation as compared to all the other indices. And what we want to use is actually a cutoff based on the study. We saw that a cutoff value of 184 degrees was best at actually predicting type 2 diabetes. So patients with type 2 diabetes were 4.2 times more likely to have this angle index of 184 degrees. And finally, just putting it all together, we would want to know where the umbilical waist to hip ratio comes in. It just matches that with angle index. Now remember, the reason why we are not using umbilical waist to hip ratio in our analysis because it is sex dependent. However, angle index is actually sex independent. And so these results and these analyses that we did, we published it in BMJ Open. And so we have this angle index model published, and the conclusions of this study can just be summarized in two main points, which are that patients with type 2 diabetes have a higher angle index as compared to controls, and angle index appears to be a superior sex-independent anthropometric index in relation to type 2 diabetes. So I'd like to thank everyone for listening to this presentation. And of course, I would like to acknowledge the assistance of these organizations who tremendously assisted in this project. So thank you very much.
Thank you, Dr. Gavaskar, for your presentation. And now this is the time for intervention of the lectures. And I would like to call upon Professor A.K. Dash, Professor Balasubramanian Narasimhan, and Professor Ajay Sharma, Professor Randeep Singh Dahiya, sir, to please come on the stage. I request Professor A.K. Dash to put his opinions and convey the questions to the speakers.
Well, thank you all. It's too late for us, but too early for you guys. But great presentations, great speakers. So it's
Please,
you can see the diversity, different area, anthropology, pharmacognosy, drug delivery, and all ABC cascades, efflux mechanism, and all these things. So it's a great presentation. And if anybody has any questions, I'll be encouraging people to raise their hand, or just since not too many people, they can also use the chat functions in the Zoom sessions if they have any questions.
so far we have not received any questions in the chat box, sir. Only wishes and greetings are there.
Okay.
So,
anybody has any questions from the
the
panels? If anybody has any questions from the panels. I don't know whether Uday is here. I have a question for Professor Uday. Uday, what is the total volume of material when you are talking about this particular area? So, how much maximum volume of liquid you can inject?
Yeah, I think it's most likely the clinical doses are less than 100 microliters. I do not remember exactly how much they're dosing. I think it's less than 100 microliters for sure at the moment. But experimentally speaking, the injection of air long back, the volume was 1.5 ml, but that was with the intention of deforming so that retina is detached and you want to bring the two together. So you have to push from behind and bring them together, right?
Yeah.
So the largest volume injected was there. And also there's a pressure. You cannot easily inject a large quantity of volume. Uh, I do not recall exactly what was the clinical volume. I think it's less than 100 or less than 100 microliters in the clinic, because there's one product at least that has been dosed up to phase 3.
And since we are applying pressure, I think viscosity of the liquid also is very important. So you have to be careful because of the viscoelastic properties of those fluids. We should be very careful in those injections too, I believe.
Correct. Yeah. I mean, that's where the device engineering becomes important for that space, because even if your needle is slightly longer, you can go up to a deeper region. You can break a vessel. That's a possibility. Um, and then the sclera thickness is different in different locations. So all those things have to be considered. But that's where the design of the needles will be such that as soon as you see a resistance change, then you release the medication, meaning you understand the resistance of the tissues, and when the resistance changes, you can release the medication. That's the kind of design that will be most useful.
Thank you.
Chair, I have a question for Dr. Tiwari. So, Dr. Tiwari, your synthesis of cyclic peptide involved solid phase technique. So why you are not preferred solution phase technique? It is quite cheap in comparison to solid phase. Can you highlight some advantages of that? Thank you.
Dr. Rakesh Diwani, please unmute yourself and talk.
Yeah, can you hear me?
Yeah.
Hello?
Yeah, here. Yes, sir, you're audible.
Yeah, sure. Thank you. So basically, a solid phase is preferred because you save a lot of purification steps. And for these initial stages, you can make a peptide pretty faster, and there are automated peptide synthesizers. So they save a lot of time. And for, the purification part, that is much easier with solid phase. And that's what we do in early phase of drug discovery. Yeah. Once we are with the product, like, going for clinical trial, we need, in gram quantities, then we go to the solution phase. Yeah.
You have prepared a chain of several amino acids. So whether you required any protecting or deprotecting agent in those? Can you highlight that?
That's a good question. So basically, all these amino acids, we purchase them as a FMOC TBU protein group. So FMOC is a amino terminal protection, and TBU or titile is a side chain protection of amino acids. And then only acid group from the amino acid will react to the amino group of the previous, previous or solid phase amino acid. So by doing that, we have only one functional group at a time for chemical reactions. So side chains are protected. Once the synthesis is done, like all the amino acids are assembled on solid phase, in the last step, we remove all the protecting group and the polymer and resin, everything. And then peptides comes through the solution phase, and we purify it by HPLC.
So then it is a mixture of both techniques, solid and solution phase, that means.
Yeah. At the last step, it comes to the solution phase.
If specifically you want to mention, if FMOC we attach to NH2 group, then in removal, what is the procedure? How to remove that?
So we use a 20% piperidine in DMF. So piperidine is used to remove the FMOC group, and this is a mild base. And basically, we use this to remove FMOC. And there was a Merrifield synthesis who used basically high acid, like, hydrogen fluoride, HF. and TFase to remove the protecting group from the amino terminal and the side chain.
Is it a hydrolytic reaction?
No, it's a radical reaction. Basically, it goes through the
Okay.
radical mechanism to remove the
Because
apple.
I work on solution phase technique, and we usually work with BOK, and then we removed it by hydrolysis with lithium hydroxide.
Or lithium hydroxide. Yeah, that's another method.
Okay,
Yeah.
thank you very much.
Welcome. Sure.
My question is to Professor Kompella. Uday Kompella. Can you hear me, sir?
Yes.
Sir, can you say something regarding why you have selected this particular target in the eye, the suprachoroidal space?
Right. I mean, there is a condition called as macular degeneration. And within macular degeneration, there's a wet form of macular degeneration. And that affects the choroid of the eye. And the choroid has new blood vessels that develop in the choroid. So that's a key target for choroidal neovascularization. So if you're treating choroid, why not approach, why not reach the target directly, as opposed to going through the vitreous and then reaching the target. But I mean, even retina could be impacted, but this provides a much closer access to the choroid at least.
So you have very good research data, sir. I have seen your presentation. It was very nice. Can you say what are the other drugs are in pipeline in your laboratory, which you are?
Right. I mean, first, the drug that, with respect to suprachoroidal delivery, the triamcinolone acetonide, which is a corticosteroid, that's the most advanced one. And I actually checked the volume, it's 100 microliters. They're injecting 100 microliters. And within our lab, we work on steroids. We have worked on other steroids. And then we also work on NSAIDs because anti-inflammatories are very commonly used in the eye. And we have modified some NSAIDs. We have modified some steroids. So those we work on. Also, we have worked on some proteins. There are a couple of proteins that we discovered. One is a fusion protein between tumstatin and transferrin, and another one is a fragment of a survival factor. And both are again useful for retinal degenerations. And the fusion protein that I mentioned is particularly useful for the wet form of macular degeneration. So that could be potentially delivered in that space, in the suprachoroidal space.
Thank you, sir. My question is, sir, next, I think so, Priyank Kumar. Sir, Professor Priyank Kumar, just, I think you are working on ABC transporters, right?
Right.
So that is for mainly for the efflux mechanism that will produce the resistance. So can you say, sir, what is the major drugs currently or you are working on, or what is the role of your this one? The drug what you have said, that AC220, I think so you have checked, right? So it is a synthetic compound or made from your laboratory or some other, it is a drug?
No, it's a synthetic compound. It's a small molecule tyrosine kinase inhibitor that is already approved. Actually, it is in phase 2 clinical trials for acute myeloid leukemia. It blocks constitutively active FLT3 kinases.
So in your laboratory, you are doing some synthetic compounds also. That is somewhere if we are getting some compound with very good anti-cancer activity, can it be checked for the this one ABC transporter property? That
yes. So that is the approach. What we do is we contact different drug companies and all who are manufacturing these compounds, or some other labs, so that we can get the anti-cancer drugs, and then we screen those drugs for their activity towards any of these pumps. So basically we focus on MRP7, MRP10, MRP1, that is ABCP1, and BCRP protein, that is ABCG2.
Thank you, sir. Tiwari sir, good morning. You have very good. I have seen your presentation on the peptidomimetics, right? You are working on the peptidomimetics for the beneficial of developing antimicrobial drugs.
Yeah.
So what are the other areas, sir, we can explore in this?
So basically, this cyclic peptide platform, we use it for coronavirus even. So there are some compounds, they are very good in the coronavirus. So, but the problem is that they are not safer because they work on the membrane. So selectivity of these compounds is a challenge. And this is the place where there are only one FDA approval of daptomycin came out. There are several clinical trials failed out. So this is a challenge with this series of compounds. So, but other than that, we have some new generation of compounds we are working in lab, and some others are like the cell penetrating peptide for delivery purpose for delivering anti-cancer drug. So there are a variety of projects going on in the lab. So if you have any specific query, I will, happy to answer or collaborate if you have anything in mind. So.
No, actually, what I'm saying, we are also working in the area of antimicrobial as well as anti-cancer. So in our laboratories, our student, they are synthesized and we used to check for anti-cancer activity. I have a collaboration with Malaysia. So the best molecule, can we go for this further development either using, but we are not working with the peptides. That's the main problem. But I think so, Priyank Kumarji, can I ask that, can we go for some, if best molecules are available, can we go for some collaborative research that where we can go for this ABC transporter property? Can we check it?
Yes, definitely. We can talk more about it offline, and I'll be more than happy to, collaborate with you on that.
Thank you, sir. One of my question is to Professor Susan. Ma'am, you have very beautifully summarized the Caribbean. What are the plans or traditional uses? So, what is your future focus? Or you are focusing on some particular region in Caribbean region? You are focusing, right? So, what are your future targets are your objective in this field?
Okay, thank you very much, Professor Balasubramanian. We have just started the news chapter in the West Indies chapter and just getting now connected with a pharmacist. We would like more professionals to join us here because there's a lot of research. Caribbean people are so much into bush medicine. So we want to know what are the, like hypertension is number one problem here, obesity is a problem, diabetes is problem. So a lot of people are using herbal medicine, but there's no scientific, background to support their usage. And most of the time the dosages are a lot of problem. So we would like pharmacists to help us, join us to, analyze some of these traditional plants. And then there's one research that was going that was left incomplete, that was on shadow benny. It is a very endemic plant to this region, and it is used in a lot of things. So we want to know and understand the phytochemicals and how it can be extracted for drug use, especially for hypertension. So there are a lot of open areas for research. And if people are interested, they can join with us and analyze some of these phytochemicals in these plants, which are endemic to this area, and use it in the in the pharmacology and in the region. So that's where we want to head to, but we can discuss further in that matter. Thank you.
Thank you. My last, I think so, Dr. Gavaskar. You are also, I think so, very have a young researcher, and I feel you have did a lot of work as him. Sir, apart from this, this type 2 diabetes, that is sex dependent angle you have that you have studied, what are the other areas which you feel to be focused in the area of type 2 diabetes? or which you are in your?
All right. So in this study, what we did was we looked at a specific gene, a single nucleotide polymorphism, and so we expanded on that in another study. So the research is basically completed, is just to publish it. And so we saw that there is, apart from, because type 2 diabetes is a multi-genetic problem, so it's not one fixed gene that causes the problem. So now we actually found another single nucleotide polymorphism that is related to type 2 diabetes. And so the more data that we collect for these single nucleotide polymorphisms, is that the greater the predictive model for type 2 diabetes. And the reason why this is important is that we need more researchers looking at these particular single nucleotide polymorphisms, because eventually the data would be collated. And remember, we are moving on to the phase with quantum computing. So a lot of data can be used, and eventually that is the future of medicine. So once you go to a hospital facility, what we eventually foresee is that your blood, and it will examine your actual DNA, looking at those those single nucleotide polymorphisms, even combined with certain indices, maybe angle index, maybe some other type of anthropometric index, and that gives you a better predictive model for type 2 diabetes, and thereby you can start better ways of treatment and preventative medicine for that condition, even for obesity as well as hypertension.
Thank you.
Yeah. Thank you,
Dr.
Narasimhan.
Gabashkar, I have a question for you. You have 140 patients, I believe. Have you taken into account metabolic disorder of those 140 patients? Do you have any metabolic disorders or metabolic syndrome?
Yes. So what we did is that we actually weeded out the persons with those with metabolic syndrome. So we have the data, but it's that we didn't want to include them because we didn't want to confound the end results. So we strictly restricted it to a control group, and those with type 2 diabetes. Because if we put in those with the metabolic syndrome, it could skew the data, and so we would not entirely know. But what we did, we also performed a separate cohort with the study. That data has not been published as yet. So we're looking at those with metabolic syndrome in relation to that of type 2 diabetes. But again, we are seeing that if you have metabolic syndrome, if you have type 2 diabetes, this angle index, this AI of 180, once you plug it into the formula, once it hits 184 degrees, it is indicative of ill health, no matter whether it is metabolic syndrome or type 2 diabetes. So it's something with that angle. And the thing is that what we did was that we include hip data as well as minimal waist hip ratio and that of umbilical waist hip. So what this does, it eliminates the error of whether you use minimal waist or umbilical, because we incorporated all in the formula, and thereby we provide a specific index that is similar in the index is not dependent on sex. So that index is similar to that of HbA1c in that you don't discriminate with sex, unlike waist hip ratio, which is discriminatory because you have to use different cutoff values for females as compared to males when considering a particular comorbidity.
Thank you.
You're welcome.
Thank you, sir. Thank you, sir.
Professor Susan, I have a question for you.
Yeah, I request Professor Ajay Sharma, if you are driving, please park your car and ask. If you are sitting in the car, that's not a problem for us.
Yeah.
If you are driving, please park your car on site and please intervene.
I have a question for you, Professor Susan. Yeah, can you hear me?
Yes, I can hear you, sir.
Yeah. Did you come across any of the medicine which is effective against RNA virus or which shows ACE2 inhibitory activity?
I didn't hear the last part. The virus?
ACE2 inhibition.
Um,
Uh,
can you say it in a simple language? Because I'm an anthropologist, I'm not a pharmacist. Yeah.
no, any ethnomedicine against RNA virus?
No, I haven't come across as such in my research. So
Okay.
there's still a lot to be done, and we haven't gotten to that point as yet. So that is where we would like more pharmacist people to join us and give us more direction into where we could, what kind of plants that can be analyzed to see its value in the drug and
Hmm.
to see its value in bringing down because Trinidad has it's actually hitting number one in the region for diabetes among the East Indian population, and obesity is very high. So a lot of people are resorting to folk medicine, to home remedies, which are not proven scientifically, and it can be dangerous because the drug dosages can be harmful. For example, moringa. People are using moringa like anything. So there are moringa tea bags in the pharmacy. There's moringa tablets. There's moringa everything. So people are using moringa to bring down the diabetes. So if they have diabetes, they are using moringa. And the next one that has come also in the market is turmeric. So turmeric capsule, moringa capsule is not prescribed by any doctors, but they just go to the pharmacy, buy, and they self-administer themselves. So different those things will work differently to different people because of the different body composition. So if I make a moringa tea for myself, I'll put a handful of leaves and just boil it and drink. It will work differently for me than for another person. So yes, moringa helps, but how much of moringa, how much time, or and then based on your age and any pre-existing conditions, because if I drink moringa, my sugar level just comes down, it just drops. So it's not good for me to drink. So a lot of issues are there. People are using as home remedies, but they don't know the safe dosages and it can be detrimental. Okay, so just an example I wanted to share because of the condition, there are diseases that are in high and people want to control it. Of course, the health ministry is also made the drugs free of charges, metmorphin and stuff like that. Dr. Ramanan will agree with me. So a lot of things are in this region that still needs to be explored, and we will need your help, assistance, and your research to help us move in that direction.
Okay. Thank you so much. And one question for Professor Compella. Sir, what about the bioavailability status of the drug that you have administered via suprachoroidal route?
If the target is the choroid, then
Yeah.
nearly all the drug or most of the drug is available. Most of the drug is available right at the target. And so by becoming invasive like suprachoroidal, you will improve the availability in the back by a lot compared to your eye drops or other non-invasive modes of administration.
Thank you so much. Thank you. Over to you, Jayabalan, sir. Thank you.
Thank you very much, sir. I think all the chairpersons have asked their questions. I need one clarification from Professor Uday Kompella that I have typed also. Sir, can you please throw some light on the drug that can any drug be used for uveitis through this suprachoroidal route of administration?
Yes, I mean the corticosteroid that I mentioned, triamcinolone acetonide, the indication is uveitis. So that's what it is being developed for, and it has been. I mean, NDA has been submitted. They're revising and resubmitting the NDA. I'm not sure where exactly they are, but the indication with the corticosteroid is uveitis, and I'm sure more can be developed for that particular indication, especially the posterior uveitis.
Okay. Thank you very much, sir. Thank you. Thank you. And I think all the speakers have given their valuable talks, and the chairpersons have intervened all the speakers nicely. And the first session of today and overall fourth session of this convention has been successfully completed by this time. And by saying thanks to all the speakers, chairpersons, and the president of APP, secretary of APP, and my organizing team colleagues, I'd like to end up this scientific session 4 of APP 9th Annual Convention. And
Prof. Jabalan, you can also invite all in the valedictory function.
Yeah. Sir, this is our program today, as I already conveyed. The second session will, the fifth scientific session, second session for today will start by 11 o'clock IST, and that will go up to 2:55, and from 3 to 4 we are having e-poster presentations by students and teachers, and there are around 55 abstracts which has been received and processed in the e-proceedings, and now we are getting the posters from the students. So far we have around 25 e-posters that will be presented from 3 p.m. to 4 p.m. IST. And after 4 p.m., we have the valedictory function of this 9th Annual Convention. And we request the presence of all the speakers, chairpersons. The invite has been sent to everyone. You all are welcome. Please come and attend this valedictory function and bless us for further conferences. So with this invitation, and sir, do you have any message to convey, President Dr. Daya? Please.
No, nothing much to share. Although the discussion was very productive, I think it will be helpful for the research in different research areas. And I am highly thankful to our scientific chairpersons as well as the resource persons of the session. And I'm waiting to welcome you at the valedictory function. Thank you, sir.
Yeah, thank you. And we'll start our session by 11 o'clock, the fifth scientific session of the 9th Annual Convention. Thank you then.
Thank you all.
Thank you.
Thank you.